Welcome everybody here to the ISB. I'm Jim Heath. I'm the president of the Institute for Systems Biology. There's only one star of the show today, of course, and that's Mary, whose great 2001 Nature Genetics paper changed how we think about the immune system. Just a few words about ISB. We're an institute that was founded in 2000 by Lee Hood and several of his colleagues, Rudy Aebersold, John Aitchison, and Alan Adiram. And about a decade ago, we partnered with the Providence healthcare system. We have an alliance with them, and that really informs many of our programs, which are grounded in fundamental science and fundamental human biology, but also translate into clinical effectiveness to transform healthcare. And with that, I will pass it over to Lee to say a few words.
Well, I'll give you a little context of Mary's Nobel Prize. I first met her back when she started working with Darwin Molecular, a company we started here in Seattle in 1992. And the vision of Darwin Molecular was you could use genomics and genetics to actually clone genes. And I'll tell you, when we started the company, there was no genome that we could have a lookup from. You had to do it from scratch. And it was an extremely challenging idea, and there was enormous skepticism. Well, Mary led a team that approached this by using mice as a genetic tool together with DNA sequencing to carry out the assays and everything. And I think what was really remarkable about Mary is she went through two fusions. Darwin fused together in 96 with Chiroscience, or 94 with Chiroscience, and then fused again with Celltech in 96 or whenever it was. So what I think was really remarkable about that is she held the team together, and she was able to use indeed genomics, the sequencing, the mouse genetics, and of course she had to use systems thinking because you had to integrate all of those different pieces together and everything. And that led to the cloning of a gene called FOXP3 that gave fundamental insights into this whole aspect of human and animal immunity called peripheral tolerance. So I think Mary did three things. One, she really validated the idea, the power of genomics and genetics. I think two, she demonstrated beautifully how powerful team science can be in putting together all the different people. But she, of course, had to apply a systems approach and bring it all together. But finally, she made a discovery that really transformed a fundamental aspect of modern immunology.
Okay, so thank you both, Jim and Lee, for a good lead in there. So as Lee mentioned, I came to work at Darwin Molecular at the end of 1994 and was so excited I was fresh off my postdoctoral work in Toronto and was so excited to find a place in biotech as opposed to an academic position where there was this very strong belief in using, as Lee said, genomics and genetics to do something really important in the world of drug discovery and, eventual treatment of human disease. So early on, I want to acknowledge that my coworker and co-winner, Fred Ramsdell, was kind of the other half of the story where he and his team brought, enormous immunology expertise, which I did not have at the time. My background is really in, mouse genetics, molecular biology, genomics. But we made a great team at this small startup company where we were given, the freedom to try different things. And it was a really exciting time because the Human Genome Project was chugging along and producing all these amazing results that were not, it was still early days. And so we had to do a lot of work in parallel that was similar to what people were doing in the big sequencing labs. But more specifically for this story, we collaborated with the Oak Ridge National Laboratory where this very interesting mouse mutant was discovered that had a really profound defect in its immunological response. And so it had a massive kind of autoimmune condition, which is lethal within the first couple weeks of life. It was pretty clear early on that it was located on the X chromosome, so only male mice were affected by it. Females could carry, one copy of the mutated X chromosome with no discernible phenotype. So the normal copy of the X chromosome was sufficient to keep them healthy. So it was known to be on the X chromosome. There had been some initial mapping that sort of had given people some clue as to where on this very long chromosome the general region where the locus was. But if we really wanted to identify the real molecular basis for this interesting phenotype, we were going to have to do a lot of genetic work first before we could dive into really analyzing things at the DNA level. So I was able to initiate a mouse genetics program that allowed us through breeding and identification of recombination breakpoints on the chromosome and whatnot, we were able to narrow down the location for this gene, presumably one gene. It seemed like the hypothesis was that the gene would be a loss of function based on how things were inherited and also what the phenotype was like in female carriers. And so we used every resource available to us at the time. We were any new genomic sequences that were deposited into databases, we took advantage of, human and mouse. There were a number of other sort of mapping resources that were available at the time. And it was an interesting time because a lot of those resources were coming out of academic labs where, their funding would have been federal funding, but we in this biotech, we found ways to be able to also take advantage of those really valuable resources. Anyway, long story short, and it was a really long story, these kind of projects were very time intensive and labor intensive, but I think we were able to make very smart use of the resources that were available to us at the time. And we also had a very strong bioinformatic team as well that helped us sift through the enormous amount of DNA sequence we were looking through. So we were able to narrow in on a locus on the X chromosome. We found it was about 500,000 base pairs long in the end, was the starting point for really doing a deep molecular analysis of the region. We identified a number of genes in that region, and many of which were completely novel and not, they hadn't been described before. And then it was just a whole lot of sequencing to compare normal X chromosome to the X chromosome that's coming from the scurfy mouse. And eventually we found a very small, it turned out to be a 2 base pair deletion in one of these novel genes that became the FOXP3 gene. So it is a member of a large gene family of DNA binding proteins or gene products. And at the same time, Fred's team in the immunology group was looking at, more of the cell biology and the immune function in these mice. And once we had the gene in hand, then we could make even more hypotheses to test as to how it was functioning in the cells. And ultimately, it led to really new insights into a very exciting and previously unknown piece of the immune system, which is how is the immune response regulated or tamped down. There'd been kind of hypotheses around the existence of a regulatory component in the T cell part of the immune system. And it turned out that FOXP3 plays a really important role in regulating that function in those kind of cells. And I'm very happy to have Jim here, who can really fill in the holes on explaining better kind of where that part of immunology has led. So this was a really early discovery, but the impact has been felt over the couple of decades since it was first described by us.
Okay, so I'll try to take you up to present day on the impact of this. So, and at the turn of this century, around the year 2000, there was I guess an outstanding mystery on, there's many autoimmune diseases where your immune system turns against you, begins attacking your own body. And the outstanding question was not why does that happen, but why doesn't it happen? How does your immune system maintain a level of tolerance that allows you to eat food and to have lots of foreign things introduced into your body, but to selectively attack the things that, like a virus or something? There had been hypotheses that T cells, there was a regulatory type of T cell, but it was the kind of hypothesis that if you said it publicly at a conference, you would certainly not be asked any questions and you might be shown the door. And what Mary's and Fred's discovery did was to actually show that there was a very important gene called FOXP3 that is a hallmark of what are called regulatory T cells. And so since that time, this discovery has had an impact in virtually every aspect of human health and disease. So in cancers, you actually would like the immune system to attack a cancer, and it oftentimes doesn't, because of these regulatory T cells sit there in the tumor and suppress the immune response against the cancer. And in fact, I think 4 years ago, the Nobel Prize was given to Jim Allison and a couple of others for cancer immunotherapy. Jim's fundamental discovery of this drug that targets a molecule called CTLA-4 actually knocks out the ability of CD4 Treg cells to stop the immune response against the cancer, and it opens up the floodgates for the immune system to get rid of the cancer. On the other hand, you have things like graft versus host disease, things that are associated with organ transplants and things like this, where you would actually like to utilize the regulatory aspects of the immune system to allow your body to be tolerant of a newly introduced organ or some sort of transplant like that. And that's basically where the frontier of regulatory T cell biology sits right now. People are developing therapies that allow you, based upon this fundamental work, that allow you to think deeply about how do you promote immune system tolerance selectively in cases where you want it, and how do you keep it at bay in places where you don't want it, such as cancer. And in fact, Fred's Ramsdell, who's also a good friend of us, is working he's chief scientific officer at Sonoma Bio, and that's one of their major programs right now. Okay.
We open it to questions, or?
Well, maybe you should comment on how you found out that you got the Nobel Prize.
Well, first of all, the last 24 hours or so have been really amazing, really overwhelming, and quite exciting. Probably don't have to mention that, but this came as quite a surprise to me. So this work, as Jim said, the paper came out in 2001, and we started the work really in 1995. But my own career, has gone different directions since then, and I really am not in that field so much recently. And so this was, oh, sorry, sorry about that. So in the very early hours of Monday morning, I was in bed asleep, and my phone was next to my bed, and it vibrated, and I thought, usually I sleep through that, but I woke up, looked at it, and there was a phone call with this strange international number, and it said Sweden, and I thought, well, that's impossible, that's spam, obviously. And I inactivated and put the phone down, and a couple minutes later it beeped again, and it was like, well, that's quite annoying. And so I put the phone on do not disturb and went back to sleep. And kind of unbeknownst to me, the same thing was happening to my husband. His phone was also going off, and he did the same thing. He turned it off. And then at around 3 a.m., so those first calls came in at about 1:20 or something, and at 3 a.m., I was aware of some noise in the house, although the dog did not go crazy. Normally, if there really was a noise, she would be barking. And then I heard voices, and it turned out that my husband had gotten up because he had heard, it sounded like somebody coming up the front stairs. And there's a young woman from AP, a photographer, at the door, at the behest of her boss, who phoned her at 2 a.m. and said, you must go to this address and get pictures of this new Nobel winner. And so she and my husband are yelling at each other through the door, like, who are you? Why are you here? And the only way he would let her in the house was she somehow convinced him. Oh, well, she had the press release or something from Sweden on her phone. She could hold up to the window and say, your wife won the Nobel Prize. And she knew she was not supposed to be the one to make that announcement. But like I said, it was the only way to let her in the house. So that is how I learned, so then the big overhead light turns on, and my husband's telling me I've won a Nobel Prize, which was pretty unbelievable. But over the course of a couple hours, it started becoming apparent that it was not an elaborate scam. That really would be too much work for people. And I spoke to the gentleman, two different gentlemen from the Nobel Committee, and that was reassuring, but again, pretty overwhelming. And the rest of the day was pretty awesome. And the support and interest that I've got from my colleagues here at ISB has been overwhelming as well. It's really wonderful. It's sad to say that the company where we did the work, Darwin Molecular, that became Celltech, shut its doors in 2003, or I would still be working on that project, because it really was an awesome time and an awesome team, and we knew we were doing something really important in the field. So, but folks like Jim Heath and other immunologists around the world have benefited from the discovery, and I think it's really opened a lot of doors in the field of immunology.
Okay.
From Lisa Eisenhower, can you say more about how ISB's affiliation with Providence has advanced vital research such as that of Dr. Brunkow?
I'm gonna let you answer that.
Oh, I can. So the question was, can we comment on how our affiliation with the Providence system has advanced vital research? Mm-hmm. I would say it's happened in multiple ways. I think one of the major examples that actually connects to Mary's research, her discovery, is right at the start of the pandemic. I think everybody recalls that Seattle was one of the first and hardest hit places. We launched a very aggressive study that actually became a model for how to study COVID and ultimately long COVID in March of 2000, before it had hit the rest of the the country. Providence, Jason Goldman at Providence worked with us. We filed an IRB to do the study on Friday. We had it launched on Monday. We were recruiting patients. That led to a lot of the deep science on long COVID, including the role of regulatory T cells in that study. And it also set the stage. We published a paper also with Providence in 2022 that really set the stage for understanding long COVID. It was the first major paper on long COVID that said you could actually anticipate long COVID in patients before they had developed it. That's one example. There's a number of oncology programs here. There's brain health studies that Lee has done through the Providence system and that Mary has worked on as well. And then many of the technologies we developed here, which allow us to treat patients, to enroll them in studies and trials and whatever, even if they're in rural locations, have come out of here. And those are studies that we're also involved in. So really, it's had a transformative effect on us, our ability to do translational science while retaining very fundamental scientific biology here at the Institute.
You know, I'd add one additional thing. We initiated a big genome effort at Providence,
Right.
which has been very successful in using key actionable possibilities in the genome, including the ability to analyze variants that there were actionable causes that you could help patients with. And the idea of pharmacogenomics, the idea that you could identify in people genes that made them unavailable to certain kinds of drugs. And in fact, it could make the drugs very dangerous for them. So Providence was one of the first, and actually it just recently published interesting paper on those efforts, but it was one of the first healthcare systems really dipped into genomics in a very serious way.
And I'll make one last comment because we have, I think, some of the scientists here in the room. A lot of the work we do here is on the microbiome, and this connects directly to Mary's discovery. So this idea of tolerance obviously must apply to things that you eat because they're foreign. Your body goes into them, they don't generate a large immune response. That's an area where regulatory T cells play an important role, and that role has been in many ways elucidated locally here at UW. But that's a program that we've launched out in the community here at ISB, and over the next year, we hope to launch it throughout the Providence system to allow people to utilize their microbiome plus genomics and other metrics to basically ensure a more healthy lifestyle and to detect transitions to disease when they happen at the earliest stage.
We have another question from Gabriel Spitzer. It's a two-part question. Can you help us understand the difference between the discovery of the Treg cells and their function and the discovery of the gene that controls it? And the second part is, what does it mean to say that a gene controls or regulates these cells?
Well, I think the discovery of the gene itself, FOXP3, was, I mean, that was our primary goal as a kind of a first step in understanding or getting a hook into the biology that was observed in these scurfy mice. And the fact that it is a DNA binding protein sort of implies that it is probably regulating other things at the molecular level and at the cellular level. And it gave like I say, it was a hook into helping scientists kind of guide the way that they would then also understand better the more biological function of the regulatory T cells. So I don't know if that answers the question perfectly, but it was a two-pronged approach to the problem.
You kind of knew there was a functional issue. You just didn't know the mechanism of it, and you figured out the mechanism.
Exactly.
And we have one more question from Lisa. Federal funding for scientific research is being cut. Do you have any concerns that this will curtail research like Dr. Brunkow's that has led to major breakthroughs?
Well, it is true that federal funding has been incredibly important for, promoting and supporting science. And certainly at ISB, the majority, I would say, the majority of the work that we do here is supported by federal funding. So obviously any loss in that hurts. I can't emphasize enough how important it has been and vital to, progress that can be made in medicine and basic science.
The other point that you can add is if you cut off the fundamental research, you eliminate the ability to start companies like Darwin Molecular, who can really take things all the way to applications for patients and everything. So I think the issue is a challenging major one, and we'll have to see how it's going to play out in the future. It's very uncertain right now.
Yeah, I'll say one more comment on that, especially prescient this week. There have been 6 Nobel Prizes announced so far this week. 5 of them are to our colleagues here in the US. Science is the one thing that the US does better than anybody else in the world. And I think that's actually, you can't argue against that almost in any other way. It's pretty remarkable, the pressures that have been put on science over the past year. This is something we should be celebrating in this country, not not the other way around. And I think it's actually is putting a lot of stress on the system. But no, I think we'll make it through it. But it's a very stressful time in science, that's for sure. And it, again, 5 out of 6. That tells you we are really, really good at this field of science here in the US. It's remarkably how that has grown. Basically, since Vannevar Bush decided this is going to be a strength that the US had post World War II.
And I think the message to everyone is determined optimism is the way we have to approach this, and do the best with what we have and hope it's going to be over.
We have another question from Gabriel. Can you spell out how basic research like this gives you new levers to pull in developing therapies for cancer, autoimmune disease, etc.? What could the new targets be?
Yeah, I'm not the immunologist, so I'm going to let Jim handle that one.
Well, you have something.
I mean, it's very clear. It was clear in the days when we first did the work that this mouse, and I guess we haven't talked at all about how we also were able to make a direct link to a very similar condition in humans. There's an analogous IPEX condition in humans where we were also able to show that the orthologous FOXP3 gene in human can also be mutated and give rise to similar, very severe autoimmune type phenotypes. So in that sense, our very basic discovery led to a very key, understanding of a human, one human condition. And from there, the basic biology and the basic, science that's gone into understanding further, just how much those regulatory T cells are involved in many different aspects and many different conditions it has just snowballed and expanded around a lot of different labs that are focusing on different, different diseases and different aspects of the system.
I think one thing that's amazing about that prescient question is that the discovery of the FOXP3 gene has probably led to at least a dozen therapies, because you're not going to use the same thing to activate T cells in a tumor that you're going to use to try to promote immune tolerance in a graft versus host disease of an organ transplant, or to try to promote or alter immune tolerance in an autoimmune disease. And one thing that we study here at the ISB is the diversity of the human immune system. And nowhere is that more apparent in these T regulatory cells that Mary discovered. Under this and that and this other condition, they all do different things. And so out of this one discovery comes a whole industry of ways to think about human biology, ways to target it, ways to mediate the immune system to get rid of cancers or promote tolerance when you want it. So it's the basic science is the lever arm that changes the world. There's no better example of it than this.
We have a new question from Lisa Stifler. Any thoughts on the rise of autoimmune diseases globally? Does it speak to how tricky the immune system is to understand and treat effectively? Is it disappointing that the trend is moving in this direction?
I didn't hear all of.
Yeah, I can read it again. No, it's okay. Any thoughts on the rise of autoimmune diseases globally? Does it speak to how tricky the immune system is to understand and treat effectively? Is it disappointing that the trend is moving in this direction?
So the rise in autoimmune disease, I would presume that there'd be a lot of ties to changes in environment and exposures and that sort of thing, which, this basic discovery doesn't really address, but it gives you the tools to start to understand the interaction between the organism and its environment.
All right.
Thank
Thank you all.
Thank you.